Hey, college biology students. So I'm going to go through some notes on osmosis, hypertonic, hypotonic. I maybe kind of mentioned this just very briefly at the end of our last video of notes. But we're going to talk about how does water move through the membrane, and you did kind of a little lab with that already. And we talked about that water does move through cell membranes, and you did this dialysis tubing kind of little cell setup that you did, and then we kind of looked at what things we thought were moving through that membrane. So that was a simulated cell. So osmosis, again, movement of water through the membrane, and this is a type of just passive diffusion. So it doesn't really take energy for water to move through the membrane. It just flows from an area where there's more water to an area where there's less water. Nature doesn't like that concentration gradient, so it's always trying to achieve this equilibrium status, or equal amounts of water on both sides of that membrane. So osmosis, movement of water through the membrane. Hypertonic is an environment that attracts water, or we say water loving. So when we had our little dialysis tubing, cell, some of you noticed that your dialysis tubing cell looked a little fuller that second day that you looked at it. And that's because inside there were higher levels of solutes inside of that cell than there were on the outside. So water moved into our dialysis tubing. So just a quick reminder of what that looked like. So if this is our dialysis tubing cell, we had glucose and starch inside of that. There we go. So there were lots and lots and lots of molecules inside of this. And on the outside in our beaker, not so much. There was water in here, and then we had a little bit of iodine, and that was kind of it. I don't know why my board is blinking, I apologize. So water wanted to move in to dilute this until we actually had equal concentrations of water both inside of here and outside of here. Okay, so it wanted to achieve that equilibrium. And just really quickly, as you know, there's a lot of single-celled organisms that live in pond water, and if you think of the way osmosis works, doesn't it seem like maybe they would just continue to, oops, wrong slide, doesn't it seem like they maybe would continue to take water in until at some point they actually burst from that? But they actually have specialized organelles called contractile vacuoles, and I know we talk about this in 10th grade biology. So water keeps kind of seeping in through their cell membrane, and it kind of fills up in this vacuole, and when it's full, it's like under pressure, almost like a bladder, if you will, or a balloon, and then it's able to contract that and squish all the water out. So it's really the only way that freshwater organisms, single-celled organisms that don't have circulatory systems, can actually get rid of that excess water that moves in through the cell membrane. Turgor pressure refers to the pressure inside a plant cell. So remember, when we talk about vacuoles, again, vacuoles in plant cells can take up to 90% of that volume of the cell up. And when the vacuole is not full, think of the effects of the plant. What might you see? What does the plant look like? It achieves something called plasmolysis, or we also call that wilting. It's a decrease in that turgor pressure. What happens inside of the plant cell, so remember, plants have this cell wall, but then inside the cell wall is a cell membrane that still is semi-permeable. The cell wall provides structure. The cell membrane has a lot more more to do with, so this is my cell membrane, has a lot more to do with that selective permeability. And then that vacuole inside of the cell can be very, very, very large. And when this is all full of water, we say that it has turgor pressure and it's pushing, it's full, like a balloon full of air, if you will. But if we dehydrate that cell, or it is that vacuole is not full of water, it looks very different. We actually have this cell membrane becomes pulled away. I'll just redraw this a little faster. So then we get something that looks like this. The cell membrane is not pushed against the cell wall anymore. The vacuole is this shriveled little deflated balloon thing in here, and it's not pushing on anything. There's no pressure. And this is plasmolysis, or wilting of the cell, like this. I talked about it a little bit in the other video that I posted. Please watch that as well. The opposite of wilting and reduced turgor pressure, cells can actually take in so much water if they're in a super watery environment, and they continue to inflate the cell until the cell actually bursts. That is called cytolysis. So that's really it for passive transport, or transport through the cell membrane that does not require energy. I think active transport I'm going to save for another day. Maybe we'll do that on Monday.